Joining me now is Dr. Stanley Nelson, professor of human genetics and psychiatry within the David Geffen School of Medicine at the University of California, Los Angeles. His laboratory develops and implements genome-scale genetic approaches to the study of complex and rare human disorders. Welcome, Dr. Nelson.
Thank you, Susan. It's a pleasure to be here.
I want to talk about the themes in this play that we've just heard of relativity. Is there any basis for believing that the propositions of what people call eugenics have any merit whatsoever?
No. Part of the reason these things get perpetuated for very long times is that, we have all sorts of genetic selection experiments that have been done by humans, dog breeds being selected for specific traits and specific abilities and the like. So that's sort of the basis for propagating some of these ideas in humans. And I think one of the things that's going to be quite powerful about looking truly at our entire genomes is that we're not all so very far apart. There's a tremendous amount of genetic diversity within humans, but there's much more similarity than there is difference, for sure. So even taking the two most distantly related people on the planet, they're almost identical, is the way to think about it.
Is it 99% identical?
It's about 99.9%. Exactly.
Yeah. But am I right in thinking that you can see a difference between the genomes of Europeans and Africans?
Absolutely. And actually, this will be an interesting feature that people will be able to explore about themselves that they might not know about their ancestral history. And this is going to be quite possible to do. That if I had somebody who was from sub-Saharan Africa and all of their ancestors, as far as the eye can see, were sub-Saharan Africans, and I had an individual who was from Central Europe or Northern Europe, and all of their ancestors, as far as the eye could see, was Central European or Northern European, there's no question that I could very easily tell who was who. And I could do that by just looking at their genes. And part of that is that we can now look at a sufficient number of DNA variations that these differences that do exist between different people from different areas are very apparent. And that's simply because of our genetic history. That is, the subset of people who've been living in Europe are sampled from a sampling of the people who migrated out of Africa, and that their population has exploded, right? So in general, Europeans are more related to other Europeans than they're going to be to the average sub-Saharan African. And that's just simply because of the time distance between those people. That is, the average European could draw their co-ancestor back to a much more recent point than when they're trying to link themselves up into the human family with an African whose ancestry comes from below the Sahara. So we can do interesting genome explorations, and people are doing these, actually. That is, are there genes that are under positive or negative selection that are different in people who have been living in Europe for centuries versus people who've been living in Asia or the oceanic region or in sub-Saharan Africa? That is, the environmental pressures might be quite different in those locations and might be then decoded somewhat in some natural selection that's actually occurred.
But explain to us why the idea that there might be a, quote, gene that gives you athletic ability or musical ability isn't a valid notion in terms of saying all black people are better athletes.
Right. So, a simple observation of all black people would get you to that point. But the genetics of any of these traits that are not some of these Mendelian traits are very, very complex. And the individual genetic contribution to a trait are typically relatively small. So you have many, many, many different, even if one were going to live and think about that it's a genetic contribution, it would be many, many, many different genes. It'd be unlikely to be a single gene at the end of the day.
What do you see as the most interesting and challenging aspects of your field at present and coming down the pike?
I think the challenge is going to be to move towards a point where we have complete genome sequencing on individual humans and being able to put that into context within specific diseases. It opens up computational challenges. It opens up mathematical challenges. It opens up actually societal, ethical challenges in terms of how do we use this data? How do we approach it? Can we indeed do this on people prior to having the disease? And I think it's going to lead to a very different way for how we think about delivering healthcare in the U.S. That is, we can't really have a system that permits genetic discrimination, which we have right now. And I think that needs to disappear.
Well, I gather that one of the projects in your own lab concerns finding an alternative methodology to using stem cells.
Yes. I wouldn't claim this is in my own lab. This is very important work and very exciting work that's going on in a number of different centers, including some investigators at UCLA and our collaborator April Pyle at UCLA and Catherine Plath have been working on means where we can take skin biopsies. So from a given patient who maybe has a specific type of mutation, we can sample the skin. And normally, people have been doing this for decades, you can take some of that skin and put it in culture. And in tissue culture, the fibroblasts, which are the cells that kind of hold the skin together, if you will proliferate. And they'll proliferate for a little while and then die. So you can study them for a little bit. We have ways of transforming those to live a little bit longer, but they still look like fibroblasts. So if I wanted to study a muscle disease, so for instance, I want to look at ways that I can develop therapeutics for Duchenne muscular dystrophy. As an example, what I would like is I would like to actually have a muscle cell line that has my exact mutation within that cell and study it, but I'd like that cell to be a muscle cell. So how can you get to that process? The fibroblasts that are taken from patient skin can be grown in the lab and then treated with a series of different genes such that those cells revert back into a cell that looks like an embryonic stem cell. So what does that mean? It means that it's not an embryonic stem cell per se. It's a fibroblast that got induced to have these stem-like properties. And that cell can be induced with some tricks in the lab to become a neuron growing in a tissue culture dish. It can be induced to become a muscle cell growing in a tissue culture dish. It could be induced to become a blood cell. And what that means is that we can now have a source, which is very important. We can have a source of stem cells that can be induced back into the cell type that we really want to study in the laboratory with the exact mutation that people really have. So that's an extremely exciting thing.
It is fascinating, absolutely fascinating. Well, thank you so much, Stan, for talking with us today.
Well, it's my pleasure. Thank you very much.
I've been talking to Dr. Stanley Nelson, professor of human genetics and psychiatry within the David Geffen School of Medicine at the University of California, Los Angeles.